Thanks everyone for being here. I want to introduce Audrey Nelson, who started working in our lab in the summer of 2021 at the Dimensions experiment at the field station. I was talking about foliar pathogens, which piqued her interest, and pretty much the rest is history. I've had a great pleasure of being her mentor over the years, and I'm really excited for you to see some of the research she's been working on. So yeah, take it away, Audrey.
Thank you, Haley. Okay, so I'm Audrey Nelson, and my research is on the differences in disease incidence across species richness and phylogenetic dispersion gradients. And Haley Burrill was my mentor, and Jim Bever helped guide the research. So pathogens are antagonists which harm their hosts, and pathogens also tend to be specialized. They come in different forms such as bacteria, viruses, and other microorganisms, and they are specialized in order to break down their host defense mechanisms. So an example of specialization would be that frog pathogens would likely not infect a plant, and a plant pathogen would not infect a frog. And so plants specifically are, they are specialized, and in order for pathogens to break through their specific defense mechanisms, they have to be specialized for each specific species. And pathogens do cause harm, but they also play a vital role in structuring ecosystems. And specifically in plant communities, they help to maintain diversity by having a large effect on fitness. So dilution and specialization go hand in hand, and because pathogens are specialists, when there's a greater variety in plant species and phylogenetic diversity, then the accumulation of pathogens is lower. So this is due to the dilution effect, meaning that when there's a lower availability of hosts, then there will be lower transmissions of the pathogens because that availability is going to be diluted. And as you can see here, this is an image of one of our pathogens. This is on sulfium, and this is sulfium blotch. So what we expect to see is that in monocultures, there's going to be significantly more disease as compared to multiple different species plots. So on the right, you can see that this is a cartoon showing a monoculture, and it is overrun by disease. And then on the left, this is a figure showing multiple species, and you can see there's significantly less disease. And then here is a figure showing monoculture versus multiple species versus multiple families. So you can see that the single species has significantly more disease as compared to the multiple families, which has the most diversity. And then these figures up here, which are sort of blocked, but they are showing the trend showing that as plant species richness is increasing, the disease is decreasing. And then over here, you are seeing as phylogenetic dispersion is taking place, the disease is decreasing. So the underdispersed is plots with species all from the same family versus the overdispersed showing multiple species from multiple families, and it has the lower disease. So this research took place out at the KU field station, and there were 240 plots and 18 different native prairie species. So there were three main manipulation factors: plant species richness, phylogenetic dispersion, and precipitation. And you can see down here on this table the different combinations. So we have the underdispersed, again, meaning that it's multiple species from the same family, and then the overdispersed, meaning multiple species from multiple families. And everything was replicated so that there was a high and a low precipitation treatment. And here is an image showing all 18 of our species. So we have three different families. We have grasses, legumes, and composites. And in each family, we have 6 different species. So in 2021, we randomly selected 8 individual plants per species per plot, and then we recorded the amount of disease seen using a 1 to 10 scale. So on the left is a level 1, and then on the right is a level 10. And in 2022, we decided to only select 4 individual plants per species. And this was because we were having trouble collecting data on 8 individuals per species per plot because there was a lack of plants, that many plants in each plot. So then we recorded using the same scale again. And then we went and collected samples of the pathogens in the field, and we sent them off to K-State laboratories, and they were unable to identify the pathogens. And we believe this is because they focus mainly on agricultural pathogens rather than native plants. So I then cultured them, and I'm hoping to perform single sequencing on them and identify the pathogens on my own. So our results showed a really nice trend, showing that downward trend, that as plant species richness is increasing, the disease is decreasing. And so this figure is showing our overlapping data from 2021 and 2022. So we saw this trend in 6 species between those 2 years. And also, I would like to note that we didn't see this trend in the grasses. We mainly saw this in the composites and the legumes. Here we are looking at the phylogenetic dispersion. So on the right, you are looking at the underdispersed side, and on the left, it is the overdispersed. So here we are seeing again that with greater diversity, there's lower pathogen accumulation. The overdispersed is going to be lower, showing that trend. And we saw this in 7 species between 2021 and 2022. And again, we only saw this in 1 grass. Mainly we saw it in composites and legumes. And then here is an image showing that trend that we saw. So as you can see there in both the legumes and the composites, we found that 4 of the 6 species showed significant results, and we only saw that 1 of the grasses did. And I would also like to note that in 2022, we ended up only having 17 species due to the lack of data on COEMAC. So these were the results from plating the pathogens. We plated both the fungal and oomycetes. And yeah. So our findings support the dilution effect and pathogens accumulation with low plant species richness and phylogenetic diversity. And this is due to plant pathogens being specialists. And this can help explain how dilution can help productivity increase with more diversity. And we also found that plants are more vulnerable to pathogens under high stress. And this gives us some insight into plant community diversity. So we also know that the dilution effect is only as effective as specialization is in pathogens. So if pathogens are less specialized, then the dilution effect will be less effective. And this raises a question as to what extent is the dilution effect effective in humans and mammals, or is it only effective in plants? And so as we know, biodiversity is vital for life on Earth, and it provides essential features for all life. And then personally, for me, what's next? I will be graduating in December, and I will be starting my master's at the University of West Florida in August. And I would like to say thank you to PhD candidate Haley Burrill and Dr. Jim Bever. And I would like to say thank you for the Kenneth B. Armitage Award, which allowed me to learn new skills and present my research at the ESA conference. Thank you. I'm open for questions if anyone has any. Yeah.
Why do you think you don't see the same pattern in grasses?
Our thoughts are that because the leaf surface area is smaller, I'm not sure if we're entirely positive. We're still trying to figure that out.
I'll raise your hand out.
Hi, can you hear me?
Yeah, hi.
Hi, very nice talk. I think it's really impressive, the work you've done. I'm just curious, it certainly makes perfect sense to analyze the data in terms of experimental treatments, but obviously things have changed over time with the plots. Since we have measures, I think, of the abundance of any particular species in any particular plot, have you guys tried analyzing the data towards the kind of actual presence as opposed to just the, I'm not sure I'm explaining this very well, but instead of just the experimental treatments, looking at the actual kind of observed appearance or density of the plants in the plots?
Yeah, we do have the above ground biomass data, if that's what you are asking.
Yeah.
So we are thinking about looking at that above ground biomass data. We have all the information for it, so we're definitely interested in looking at that.
Yeah, that's great. Thank you.